Individual conversations down now and start to focus in on the reason we're all here. My name is Bob Brower, and I get to welcome you with two hats. I'm currently the president of the Owasco Watershed Lake Association, and I served 3 terms on the board of this theater. So I get to be one of the ambassadors on behalf of Auburn Public Theater and welcome you to the main stage here in the theater, and we generally ask how many of you are in Auburn Public Theater today for the first time. Just curious about that. Well, come back and see a plethora of shows and entertainment and comedians and great music, and it's all online, auburnpublictheater.com. So, number of acknowledgments that need to be made. I think the theme for today is pretty clear in my mind, and that is the need we have as a community to come together, to collaborate, and to address what we in the Lake Association think of as a crisis in water quality. One that's been identified, one that we have shown a great deal of light upon, and you're going to hear more today, and if light is the metaphor, then there'll be more sunshine put on the issue as one speaker after another presents what we know, what the status of the lake is, and then we'll get into a little more detail from individual perspectives. We had a great deal of science going on, but we didn't want to leave you with the impression that it's only science and monitoring going on. It is a critical part of what's happening in the community because the science upon which we hope to put public policy is not highly developed. So we're in that messy area between what science can tell us that we can use as a foundation to develop our public policies. And there's a great deal of uncertainty around the science. And again, it's not our intent to pretend that isn't the case, but rather to put light on it so we all understand a little bit better what we do know and what we don't know. And at the same time, there are things that can be done that we know will help. We know we have to drive nutrient levels down. You'll be hearing a lot about nitrogen and phosphorus and the benefit of slowing down the loading in the lake from all sources. And then in the end, we'll focus the event on a panel that will be asked to tease out for us what they think we need to do as related to that public policy. What steps do we need to take based on what we think we know? And in between the science and that stage, we will have at least one presentation. Rick Nelson will be overviewing the actions already underway in the watershed. Because we've learned, even when you say we're here to learn the science, not to talk about what we're doing, people will leave saying, I don't know why they're not doing something. So help us understand that. Our focus is on the basic levels of what we know, and yet we will touch on the action happening, but that's a second focus for us. So it took me longer to say that than I thought it would. I appreciate your patience. A couple of acknowledgments I think need to be made. I mentioned the fact that it is a collaboration, and so I've not been able to track everybody that's here, but I know that the offices of our state reps are well aware of what we're going on. We were in Albany earlier in the week. We met with Assemblyman Finch, and we talked with our senators, Pam Helming, the new 54th District Senator that took Senator Nozzolio's place, and Senator DeFrancisco, of course, and Senator Seward's staff met with us as well. So we had a great morning. We know our state reps are tuned in. We know that our federal reps are tuned in. Congressman Katko's staff come to most, if not all, of the Owasco Watershed Lake Association meetings. Great deal of interest at the county and city level. Of course, we meet regularly with the chairman of the legislature, Keith Batman, and he's leading the county response. And then at a city level, Councilman Jimmy Giantino and Terry Cuddy are with us this morning. I don't know if Debbie McCormick is also here, but she is here. Wonderful. And Dee is here too, so we have an illegal city council meeting going on. Welcome here, welcome, and thank you for everybody being here. And there are more county legislators that I can't see or didn't see come in. Mike Didio's here all the time, a member of the Lake Association, and I guess not. Okay. At a public agency level, the Department of Health, State Department of Health, and their voice locally, the County Department of Health, the Department of Environmental Conservation are both deeply involved in efforts and part of this collaboration. So I've only to thank the members of the committee that organized today, and they do this at the same time they organize the Summer Lake Day. So particularly thanks to Rick Nelson sitting down here. Rick, would you raise your hand? Chairs the meeting of the... And if I could ask the other members of the Owasco Watershed Board of Directors to raise your hand. So if you want to throw fruit at us, here we are. And we're all volunteers doing the best we can. So my privilege, thank you for your attention, my privilege to welcome Dr. John Halfman from Finger Lakes Institute at Hobart William Smith College. Has been studying the Finger Lakes for years. We've known him most of that time, and so as always, he'll share with us the status of the lake through the eyes of his science and put it in comparison to the other Finger Lakes as well. Give us some introductory information about cyanobacteria, and then we'll keep going deeper and deeper as the morning goes on. So John Halfman, welcome, and thanks for being here again.
Hi, so my name's John. I've been looking at the Finger Lakes for a little bit while. So first of all, can you hear me way back there? Yes, no, maybe? Move this up higher. Is that better? Okay, all right. So I hate these things, by the way. But I'll try not to move it, because then you can hear me, can't hear me, and I can say anything you want, because you can't really put a sentence into there. But anyway, I've been looking at these lakes now for way too long. I should have been retired. I probably will retire sometime soon, but who knows, maybe not. I've been asked to talk about what I found in the lake last year. And what's really cool, as far as I'm concerned, every year I do this, I usually find something new. And usually every time I give a talk like this, I forget to say 10 things, but I say 1,000 others. So bear with me. When there's confusion going on in anything that I say, apparently at the very end of this morning, you get to ask me, or grill me, or do whatever you want. I'll survive all sorts of things. All right, so the photograph you see behind me is some of the sediment plumes that happened not last year, but the year before. Major plumes. Though those of you who live along the lake shore, did we have plumes like this last year? Not at all. So there was a major difference between 2014 and 15, which I'll group together, and last year. Where did most of these sediment plumes come from? Anybody know what happened to make these plumes?
Rain.
What's that? Rain. And the erosion associated with that rain. So what do you think probably happened last year if there weren't any plumes?
Rain.
Not a lot of rain. So that's the story for last year. That means I'm done. Right? I don't have to say anything else, because not a lot of rain, so maybe not a lot happened. Continuing on, I've been looking at the Finger Lakes and comparing and contrasting the Finger Lakes since 2005. That honor student shown up in the upper picture put together a reason why some lakes are really, really clean and other lakes are really, really dirty. Don't worry about the reason why right now. Focus more on the fact that if you look at Owasco Lake, it was one of the worst in terms of water quality. That got me interested in Owasco Lake. Why is it worse? And this ranking is based on the parameters that you see up there. Since that year, I determined that analyzing for bacteria is way too expensive. I haven't done it since. And the rankings change when you take one of those parameters out. So Owasco isn't the worst anymore, but it's pretty close as we go through all these different lakes. So first in 2006, when I was out there looking at lake samples, if you go out into the open lake, all these sites from D all the way up to A are out in the open lake. Notice that shown here is total phosphate concentrations. They're relatively the same. That year you learned most of the lake is pretty well mixed. Where it was different though was the last 5 sites that you see here down with these bars. Some of these bars are significantly above the top of the graph, where the top is at 20 microliters per, or 20 micrograms per liter. Some of those concentrations, mean concentrations, went above 600. Those were all clustered down near Owasco Inlet. So if you had to make a really crude guess, where's the source of phosphorus coming into the lake, what would you say? Probably down at the southern end where the main stream's coming in, right? So with this data, you go to someone like Senator DeZolio, say, hey, look, I have a good idea where all the nutrients are coming from, what's making this lake worse in comparison to all the other Finger Lakes. Give me a bunch of money to do something. I should back up a minute. That first 2006 study was funded from the Emerson Foundation. Their seed dud money has now since allowed me to come back to this lake using Cayuga County money every year since, which is great. It initiated something really, really good. Now, if you look at 2007, with this extra money that you get from Senator DeZolio through state funds, notice that it's the same sites. What do you see that's missing at the right-hand side of the graph? There's no spikes. It's almost like someone turned off that supply of phosphorus. So if you look at the difference between the 2 years, looking at some of the streams in 2006, here's Owasco Inlet right above where it comes into the lake. Here's 2007. The scales are the same. Notice that in 2007, the amount of phosphorus coming in at the southern end was a lot lower. What do you think makes it go a lot lower? Less rain. And 2007 was one of the driest years on record. 2006 was one of the wettest years on record. It suggests that rainfall somehow takes phosphorus off the land through runoff, puts it in the stream, and then delivers it to the lake. Phosphorus, one of the limiting nutrients that you have to deal with in a system like this. There's various point sources and non-point sources that I might start talking about. In terms of a point source, there's 2 wastewater treatment plants in the Owasco Inlet. One services Groton and surrounding communities. One services Moravia and some of the surrounding communities. The amount that they stick into the stream is shown on the graph on the right. Notice that the one in Groton put in a lot earlier on. But because of data like this, the DEC was convinced to suggest to Groton, actually twist their arm a little bit, you need to reduce your nutrient loading. And they did that based on scientific data that I was able to collect. This is really cool. Continuing on, what will adding nutrients do to a lake ecosystem? Well, first you got to know a little bit about a lake ecosystem. You bring nutrients in, algae can then pick them up and grow. Usually there's a limit to the concentration of those nutrients, so the algae will grow as much as they can until the nutrients run dry. That algae then feeds other parts of the ecosystem. What's really cool about lakes is when those organisms die, whether it's the algae, the zooplankton, the fish, whatever it is, they will then get decomposed by bacteria, and those nutrients get released back into the water column. So effectively, you bring nutrients into a lake, they stay there. The recycling rate of nutrients in an ecosystem like this is above 90%. Much better than we as humans can do for aluminum or lead or anything else that we might want to try to recycle. So if you bring more nutrients into a system like this, all those boxes get a little bit bigger. Sometimes it's hard to detect because they all get proportionally a little bit bigger. And the sources of these nutrients, there's a lot. You can bring in the dissolved form if you want from fertilizers or animal feedlots or wastewater treatment plants or on-site septic systems. You can bring in raw organic matter as well. In other words, untreated waste, cow poop, erosion of soil particles that have organic matter on it. Any one of those brings in extra stuff coming into the lake, which then can be used in this nutrient cycle. Too many nutrients, then you end up with too many weeds along the shore. You end up with algal blooms. If the algal blooms can get intense enough, then you can end up with blue-green algae. And I'll probably abbreviate it as BGA. Unfortunately for this lake, most of the blue-green algae blooms have high in toxins, but I'll leave that talk to Scott because he knows a lot more about that than I do, and that I would ever want to know. Continuing on, what I did last year was basically the same that I've been doing every year. I maintain water quality monitoring at 2 mid-lake sites out in the middle of the lake. I was fortunate enough to stick a buoy out in the lake that collected daily profiles as well, and meteorological information. That buoy is shown way over there. I was also able to take samples along 2 streams, the 2 biggest streams that empty into the lake. One of them is agricultural rich, the other one's not agricultural rich. Just to compare and contrast, try to figure out where sources of stuff are coming in, sources of nutrients primarily. I was able to put an auto sampler in Dutch Hollow Creek, which last year collected daily samples so I could look at daily phosphorus loads or daily suspended sediment loads coming into the creek to see what sort of variability in the watershed might bring more nutrients in or less nutrients in. As far as the lake sites, they're shown there in red. Where the buoy was located is shown in blue, those circles on the lake map. Notice they're out in the middle of the lake. And I emphasize that because out in the middle of the lake, I don't see blue-green algae in huge bloom concentrations like you do in some near shore locations. I see blue-green algae, but not in huge concentrations, okay? All my work is done from this pontoon boat. Some of the data that I collect, it's called CTD profiles. Here's the surface of the lake. There's depth going down. Here's both sites. So there's temperature. The 5 times that I went out, I do monthly samples through the summer. Here's the salinity of the water. I'm down with water depth. The dissolved oxygen probe unfortunately broke, and I didn't have enough money to replace it. Here's the light availability in a logarithmic scale. Here's the amount of algae. Each of the day, notice the algae are way up at the surface. Here's water column turbidity. One site seems to reflect the other one pretty well. Last year, without a lot of rain, I'm labeling it as a dry year. You compare it to a wet year, you'll notice the biggest change between years is there's a lot more turbidity in the water column brought in by streams throughout the entire water column. So that's shown, make sure I press the right one, that's shown over here. A lot more turbidity than that year and also this year over there. Major difference between what's going on from one year to the next. The other thing that I collect is Secchi disc depths. If you don't know what a Secchi disc is, it's the device over here. It's a round disc. You lower it down through the water until you can't see it, and you measure the water depth. Unbelievably simple tool, but unbelievably powerful tool in that the deeper it goes, the less algae is in the water for this lake. If it's really shallow, you got a bloom. If it's really deep, no algae. Really deep, like 30, 40 meters. You guys don't go that deep, but anyway, notice these are annual numbers. It's box and whisker plots where essentially what's going on is 50% of the data falls within the box. That green thing is the mean. There's the shallowest depth. There's the deepest depth. So if we go from shallow down to deep, looks like 2016 was a little bit deeper than the preceding 2 years. It suggests that there was more algae in 2014 and 15 than 16. And if you go back through this record, whenever there's more algae, you can correlate it to more rainfall, bringing more stuff in, more nutrients into the aquatic ecosystem. Less rainfall, 2016, clearer water for the most part, out in the middle of the lake. I know some of you right along the shore experienced very intense blue-green algae blooms. I'm not right at the shore. We'll have a talk later on, right after mine, talking about near-shore stuff, and I'm interested to see what he finds. Offshore, this is what's going on. You can compare it to the amount of total phosphate that's there, total phosphorus being the limiting nutrient. If you run out of phosphorus, there's no more algae can form. And if you go through time, notice that 2 high years, 2014 and 15, it then went back down a little bit in 2016. There wasn't as much biomass, algal biomass, in 2016 than there was 15 and 14. And that's a rainfall signal as well. You can say the same thing for chlorophyll, but if you look at all the indicators, whether this lake is really productive, eutrophic, or not productive at all, oligotrophic, or somewhere in between, mesotrophic. Owasco Lake has been bordering mesotrophic, oligotrophic now for some years, it's mesotrophic. Some years, it's oligotrophic. And the boundary between the two usually depends on more rain, less rain. More rain, more nutrients, mesotrophic. Less rain, less nutrients, oligotrophic. So last year, oligotrophic, based on 3 out of the 5 parameters that will define if it's mesotrophic or eutrophic. It's boundary line, right at the border. If you look at the comparison to other Finger Lakes, and what this shows is, going back to that original ranking that I talked about earlier, and it's averaged all the ranks since 2005, and put that average here in the blue bar, excluding the last 3 years. So there's the average over the past, since 2005. Here's 14, there's 15, there's 16. Water quality ranks. Worst ranks go high. So you can notice in just about every one of these lakes, worse water quality, 14 and 15, no matter what Finger Lake I go to. So there was a regional impact going on in all these lakes. More rain, more nutrients. The scale might have been a little bit different in that Canandaigua, Keuka, Skaneateles Lake are significantly cleaner, consistently, proportionally, than some of the other lakes that are there. Seneca, Cayuga, Owasco. I'm not going to talk about Honeoye because there's people from Honeoye here and they don't like me talking about their lake. Reasons why. I had another honored student try to compare those rankings to anything that might change in each one of the watersheds, whether it's the size of the lake, the depth of the lake, the volume of the water, the surface area of the lake, the surface area of the watershed, the ratio of the watershed to lake surface areas, anything she could think of, the number of people living in the watershed, the amount of money as tax base in the watershed, etc. The thing that correlated the best was the amount of agricultural land. More agricultural land, usually the greener the lake. We'll get to reasons why here in a little bit. Continuing on, looking at the plankton distribution was basically the same, out in the middle of the lake, mostly diatoms. There are blue-green algae. And there's always been blue-green algae in this lake, but never a bloom like it's been doing in the last since 2014. Never, that I know about. We also, as part of FOI, purchased a fluoroprobe. And what this instrument can do, just like the CTD, is can collect real-time data going down through the water column. And what fluoroprobes will do is differentiate between major algal groups. So if you look at some of the results, all these different color circles are different algal groups. There's temperature. So blue-green algae, these open blue bars. Way at the surface, there's a few, but not very many. This increase down in the cold bottom water is an artifact of the instrumentation. Don't believe it? There's a similar sensor on the buoy. Goes high in the cold bottom water. It's an artifact of the instrumentation. Don't believe it. I know that for a fact because after this year, I took the instrument, put it in my fridge, let it accumulate data, brought it out in the lab, it warmed up, let it accumulate data, put it back in the fridge, back and forth all week long. And I got a nice linear slope. When it was cold, it showed blue-green algae, even though I didn't change what was in the cup. When it wasn't cold, there was none. Wrong. But these little bumps where the temperature's the same, that means something. So there is some blue-green algae out there. Not a lot. not like he had in the nearshore regions. I also started some preliminary work with a drone that can take pictures of the nearshore area and also out in the open water. In the nearshore area, you can see the coming and going of the weeds, the macrophytes that are there. Very spectacular in the places that had, could offer of coating the rocks, because they came and then the next day they were gone, rotting on the edge of the beach. We also were able to bring them offshore, the drones offshore, especially when the owner of the drones let me, which wasn't very often, because I'm known to crash drones in lake water. It's an expensive hobby, whatever. I wasn't flying it at the time, but I insisted that it get flown. So anyway, my fault. We were able to determine that based on the relative concentration of the blue pixels to the green pixels, you can tell the amount of algae that's in the water column. Pretty well. So it suggests that drones are going to replace me. So it's probably a good thing I'm retiring, right? You look at blue-green algae concentrations, all 4 years. This is data collected by the Watershed Inspector's Office. Very concentrated at the northern end. Not only is the blue-green algae concentrations there very concentrated, like 1,000 times more than what I detected in the open water. Also, the toxins were very high, and they got progressively higher as you go from one year to the next. It's interesting that it's all concentrated at the northern end. Notice if you look at the wind rows shown here, the dominant wind direction was from the south, pushing floating blue-green algae to the north. So that part makes perfect sense. There might be other reasons why there's persisting there and then growing in population in the northern end, but it's something to work out in future years. I'll leave my recommendations for other people to say, because they can say it much better than me. The buoy itself provides much more data than I can with my monthly sampling. If you look at the red lines, that's when I was out there with my boat. You look at the buoy data, it's much more continuous, 2 profiles a day. That's pretty amazing. So you can actually see the onset. There's cold water, and it gets progressively warmer and warmer, then gets cold in the fall again. The surface water gets less saline through time. You can see there's a huge turbidity episode in the middle of the summer, which matched a huge algal bloom, not blue-green algae, other types of algae. So on and so forth. You can see the onset of the lack of oxygen just below the thermocline and also right above the lake floor. Not as intense this year as previous years, but it's there. You can see it actually happening. That part's really cool. You can also use this data to see what the lake's condition was when there was blue-green algae blooms along shore. So on this, what's being shown is in these squares is the concentration of nearshore blue-green algae. In blue, the solid blue, is the concentration of algae out at the buoy. Doesn't mimic it at all. They're on some sort of different signals, as far as I can tell. And then in red is the concentration of blue-green algae out at the buoy. Doesn't mimic the nearshore at all. If you then look at other parameters, there's when blue-green algae was compared to, for example, water temperature. So it looks to me they like warm water, but can extend into colder water. So they wait for warm water, then they can extend into slightly colder water based on this data. If you look at the amount of light, they don't need a whole lot of light. Matter of fact, they seem to prefer when the light levels go down. Maybe because they float, they can outcompete other algae for lower light. Maybe. I'm not a biologist. Keep that in mind. I'm not a biologist. Notice here, if you look at air temperatures, it mimics water temperatures pretty well. You look at wind speeds, they seem to like days that are calm. They also seem to like days that are sunny. I'd like calm, sunny days too. Much better sampling on those days than cold, winter, rainy, wavy days. But anyway, they seem to like those. But unfortunately, for a predictive tool, they didn't come out on every sunny day that was calm. So it's hard to predict what's going on, based on what's going on. But it'd be nice to get the buoy out there again and see if some of these consistencies hold through or not. So deployment in 2017. I don't have money to put it out there yet. I'm working on a local foundation to try to get money out there. I haven't been told if I got it yet or not. It's way too late to apply elsewhere. So it might go out, it might not. If it does, it's out there in April. If not, then, oh well. Stream segment analysis. You sample streams starting downstream, working your way up, where you see jumps in concentration or jumps in the loading of that parameter. There's a point source in there, or maybe it's a non-point source somewhere in there, and you just try to figure out where it is. The two main streams, there's Dutch Hollow Creek, way up here, and then down here is the Owasco Inlet. Those are the two, and then I sample other tributaries coming into both, and so on and so forth, working all the way up. Sampling for nutrients and suspended sediments and a host of other things. I also sampled a stream over here, right by Fire Lane 20, the very end of Fire Lane 20. It's a small tributary. That's very similar to all the other small tributaries that feed along the western edge of the lake itself. So here we have all the sites that are going through Dutch Hollow Brook. Here's all the sites going upstream from downstream to upstream for the Owasco Inlet. And what I can say is the trends are basically the same as other years. I see a little bit of increase between Groton and County Line. That little increase is probably because of the wastewater treatment plant that's there. There's a little bit of increase between VFW down to Aurora, probably also related to another wastewater treatment plant that's right there. If you look at generalize what's going on in Dutch Hollow Brook, there's a steady increase as you go from upstream to downstream. That steady increase to me suggests that there's a lot of non-point sources coming in all along the stream course. It's not just one specific point in that watershed. If you want to compare this to other years, here's a wet year, slightly larger concentrations. This one mimics a wet year because 2 out of 6 surveys were during rain events. So it looked like a wet year. Comparison, you go to over here to a dry year, significantly less nutrients coming down that stream. This not being nutrients, this is actually discharge, but the two are highly correlated. Here's nutrient concentrations, total phosphorus concentrations, pretty even in both of the streams going all the way across. If you look at dissolved phosphorus concentrations over here, pretty even as well. There was one site in Dutch Hollow Creek that drained a large farm. That one site, I think, should be highly commended because if you look at the nutrient concentrations, that one site was always high until here, and then it went back to all the others. That farmer must have done something really good, and they should be congratulated. If you compare it, same with specific conductance, by the end, last year, it was also low. He's cleaning up his act. This is really good. He should be commended. If you look at nutrient fluxes, here's total phosphorus fluxes. A lot more comes down the inlet because the discharge is much higher in the inlet than Dutch Hollow Creek. Way over here, we have dissolved phosphorus fluxes. Not a lot of story to say there other than just look at the data. If you look at then the daily information from this auto sampler, quite a different record this year than previous records in that normally there's a lot of jumps in the flux of individual parameters, this being in the green bars. This is total suspended sediments in 2016. Notice there's a lack of jumps. Notice also that in blue is rainfall. Normally there's a lot of jumps in rainfall as well. Notice not much rain until you get the end of July, beginning of August, and then some rain started to happen. Notice that the stream height, shown in red, doesn't jump up and down as well, where in previous years it always would. Remember what 2016 was? A dry year, not a lot of rain. And the stream reflected that. This was one of the lowest annual loads since I started putting a sampler in Dutch Hollow Brook. Lack of rain. I noticed a few other things with those low loads. There was a lot of pools forming in the stream. Those pools then germinated a lot of algae in the stream, which is quite unusual for streams. Usually they're flowing fast enough not to do that. I openly wonder that if the rainstorms that happened in August pushed all that algae out because they went away, and then started to rot along the near shore, providing the nutrients for the blue-green algae blooms that happened a little ways later. Because the blue-green algae blooms really didn't start until after some of those first rains. They needed that input of nutrients, and it might have been the algae that was in the stream that provided that. So here's the actual data for total suspended sediments. Here's the actual data for total phosphorus. They correlate really nicely. That's because phosphorus is usually attached to particles. Where particles go, so does phosphorus in an aquatic system like this. So if you want to look for the delivery of these things, notice the delivery, the tall peaks in the green, especially this last storm in October, which went way up there someplace. Or down here in a previous year, those tallest peaks always happen during rain events. And if you total up the total amount of sediment getting down that creek in a given year, it's always more than about 90%, sometimes 95% of all the sediments come during rain events. In between, those 5 or 6 days in between rain events, very little comes down at all. It suggests that when it rains, you have runoff, that runoff picks up soil particles, then dumps it into the water. Or maybe it erodes a stream bank a little bit. Or maybe the ditches along your highways are picking up stuff and dumping it into the stream as well. Good sources of suspended sediment and phosphorus getting into your stream. To belabor the point a little bit, if you look at the annual loads of suspended sediments or total phosphorus versus rainfall, and I've restricted the rainfall amounts to the 3 months in the springtime, because that's when the ground is bare, or could be bare. There's a good correlation. More rain, more stuff gets into the lake. Less rain, less stuff gets into the lake. It suggests ground shouldn't be bare. However, we can't do that in this watershed because farmers need to have the ground bare to plant their crops. So other things need to be done. You look at a total phosphorus budget based on the inputs from those 2 streams and an extrapolation to the entire watershed. Maybe not the best thing to do, but if you're consistent, you can at least compare 1 year to the next, to the next, to the next. With that comparison, you look at all the stuff coming in, that's in the orange bars, and then you look at all the stuff that leaves by the outlet, that's in the blue bars. Almost every year there's more coming in than what goes out. You have a classic nutrient loading problem. And with that classic nutrient loading problem, the lake should be getting greener and greener, especially during the wet years. This wet year, that wet year, that wet year there, compared to the dry ones, which probably balanced out a little bit and water quality improved a little bit. The wet years, you're bringing in a lot of stuff. So what can you do? If rain events seem to be the major player bringing stuff into the lake, somehow you have to retard the erosion of particles from the landscape to then get into the stream, which then eventually get into the lake. So it suggests a number of things. There's some other sources that you need to worry about as well in terms of these sources. Clearly there's some stuff coming in from wastewater treatment facilities. I will admit there's stuff coming in, but it's probably doing the best they can under my judgment, my opinion. There's probably phosphorus coming in from on-site septic systems. They too are being stringently monitored, probably doing the best they can. You can actually calculate how much phosphorus would get in the lake if everybody on a septic system didn't have a septic system. They just pooped at the end of their dock or peed. And according to those total budgets I have, it's small, insignificant, an order of magnitude less than some of those bars. You can look at stream bank and road ditch erosion and try to hydroseed all the road ditches and armor some of the banks and the streams so that they don't erode some more. There's some evidence that some of that's probably going on. You can look at what agriculture does. There's probably a place where you can do some more work. However, I never want to point a finger at any one of the groups. I've heard way too many people say, no, it's the septic systems, pointing a finger. No, it's the geese out in the middle of the lake, which are unbelievably insignificant, by the way. No, it's, and they point to somebody else. I think that's going completely the wrong direction. I think everybody who knows their system the best, whether you're monitoring the septics, monitoring the wastewater treatment plants, maybe you're a farmer, you know your system the best. What you need to do is figure out how to reduce nutrient loading from your system. Because OWLA has money to help you out. And if we're really good at it, we probably could get more money to help you out even more. So perhaps my call should be, let's try our best to work together. Don't say you're the culprit or you're the culprit. Try your best to work together to solve this issue. Because if we can reduce nutrient loading, I'm convinced we can probably reduce the blue-green algae problem. And the lake will go back to what people remember. 2007, I hope to get funding from Cayuga County to sample the lake sites and the stream sites. Again, I hope to put that sampler at the end of Dutch Hollow Brook. That's also coming from Cayuga County. I have a proposal to a foundation to try to put the buoy out again and to do some nearshore work, because blue-green algae live in the near shore. Clearly I'm missing something because I don't sample the near shore, so I should be sampling the near shore, see what's different between the two parts of the lake. I haven't heard from any of them yet, concretely. But we'll see. With that, thanks for your attention. I'm not supposed to ask any questions yet or answer any questions yet, but at the end, you can get me as long as you want.